The overall goals of these paired procedures are to determine fat mass in C. elegans using either a biochemical or high-throughput microscopic method. This is accomplished by first knocking down each gene of interest by RNAi in 96 well plates. Once worms have grown to adulthood on the bacterial RNAi, they are fixed in isopropanol and stained in isopropanol with Nile red. Next, stained and washed animals are imaged on a high-throughput microscope in bright field and fluorescence with a GFP filter. Fluorescence is quantified to determine overall fat mass. The second procedure is a paired method for the biochemical assessment of triglyceride and phospholipid content in C. elegans, carried out using solid phase extraction followed by gas chromatography-mass spectrometry. Ultimately, results can be obtained that show modulations in fat content for each RNAi clone based on microscopic fat staining fluorescence intensity that are confirmed by biochemically determined triglyceride to phospholipid ratio.
We first had the idea for this method when we realized that Nile red, when fed to living worms, stains lysosome-related organelles rather than lipid droplets. But when used in a fixative-based isopropanol staining solution, Nile red correctly and effectively stains lipid droplets.
The main advantage of this technique over other existing methods, like feeding or other fixative-based dyes, Bodipi-labeled lipid trackers, or Carr's microscopy, is that high-throughput analysis can be easily accomplished.